Hello, and good afternoon, good evening, or good morning, depending on where you're joining us from today. Welcome to this Twist webinar titled Hybrid Capsid-Based Methylation Sequencing Delivers Highly Efficient Target Enrichment Sensitive Liquid Biopsy Approaches. My name is Sean Bate, and I'm the Field Application Scientist Manager here at Twist in Europe, and I will be moderating this webinar today. And so now I would like to introduce our speaker for today, Christy Krusma. Christy has degrees in genetics, biotechnology, and biochemistry, accompanied with more than 10 years of working experience in the biotechnology field. She is skilled in the fields of genetics, cancer biology, liquid biopsy, method development, and data analysis. Currently, Christy is leading the work of a multidisciplinary team in the diagnosis product development process with main focus on GI tract cancers. Christy, I will now hand it over to you.
Thank you, John. So first of all, I would like to thank Twist Biosciences for giving this opportunity to give a talk on this exciting topic. So as John already mentioned, then my main focus in my research is on liquid biopsies and on cancer. And this is what I'm going to be talking about today. My affiliations are with Universal Diagnostics. So this is the company dedicated on developing early cancer detection tests based on liquid biopsy. So going into the main topic in here, I think it's no surprise to anyone that cancer is a global problem, and it's a problem that's increasing in all parts of the world, with more than 18 million new incidents per year worldwide, more than 4 million in European Union, close to 2 million in US, and the mortality rates still remain high for the main cancers. So the main bulk, of course, of the cancer cases is based on high incidence cancers like breast, lung, and colorectal cancer. And the treatment opportunities are still under development for most of them. However, if you are taking the colorectal cancer as an example in here, then you can see that early detection really makes a difference in here. So if detected in an early stage of localized stage 1 or 2, 9 out of 10 patients have a survival rate of 5 years or more. Whereas when detected in the late stage, in stage 4, the survival rates are very close to 0. Furthermore, and in some of the cancers, and colorectal cancer is a really good example of that, there is a precancerous stage. So when detected in that stage and that early, the cancer can be prevented. And this is the case with adenoma detection, where the colonoscopy findings and sending patients into colonoscopy with adenomas would prevent the cancer development overall. That brings us to the good ways of actually detecting the cancer early and the methods that would be good and non-invasive for the bulk of the patients. So liquid biopsy, of course, comes into play in here, as it is easily obtained and it has high adherence rates. And it does carry at any given point the physiological footprint of the person as it is. It has abundance of different biomarkers and allows for detection that is tissue-specific, disease-specific. The applications, of course, are varied from early detection to monitoring, recurrence, treatment guidance. So different applications, different bars where liquid biopsy can come into play in here. Of course, as blood is carrying everything that's circulating in our bodies, that means that many biomarkers, then each of them has their own specific utility and cancer surveillance and disease surveillance. But I'm going to be talking more about methylation and fragmentation in this context. And why? This is because from first sight, methylation, it is more abundantly present than mutations and especially SNPs are. It is highly disease-specific and tissue-specific, so it allows for detection not only of cancer, but also telling which type of cancer it is. It is an alteration that appears very early on in cancer development. And it is a robust biomarker to detect in a clinical environment. And of course, the important part is that in cell-free DNA format and ctDNA format, it can be detected from the blood plasma. And it does have a functional activity on the gene expression. So methylated genes are getting downregulated, as well as inhibition of methylation would activate the gene expression in here. So it makes it into a very ideal biomarker for early detection, as well as for the downstream processes and monitoring of the cancer. On top of that, DNA fragmentation is becoming more and more relevant in the cancer research as well, as it is linked to the different processes and DNA fragmentation or DNA apoptosis from the cells. So it is linked to the methylation, to copy number variants, to the mutation status and changes. And similar to methylation, it does appear quite early in a cancer development and goes kind of hand in hand with the methylation signals as we see them. And it is been shown to be quite tissue-specific as well. So the open chromatin regions and frequencies are shown to be specific to the tissue of origin. And combining this kind of information, of course, would fortify any kind of detection, any kind of procedures that are applied to the cancer patients. However, of course, as with everything, there is a drawback in using liquid biopsy, even though that it's an easy material to obtain, and most of the patients are more eager to give blood sample than, for example, a fecal sample or urine sample. Then, of course, it's the limit of how many copies of the target gene do you actually have in 1 milliliter of plasma. So as you can see from here, then depending on the tumor burden or the amount of tumor DNA that you actually shed into the bloodstream, you could be presented as little as maybe 3 or even none copies of the mutated or methylated gene in 10 nanograms of input. And 10 nanograms of input is important in here because in most of the cases, that's what you have to work with when you're working with plasma and if you're working with cell-free DNA. So that makes it challenging. It makes it kind of a needle in a haystack a situation where you really have to have methodologies that allow you to take the most out of those 10 nanograms, those 3 copies or even less copies. So every fragment would matter, every molecule really matters. And unfortunately, most of the methodologies, the conventional methodologies, have their drawbacks when dealing with such low amounts. So, for example, if you're looking at methylation and you need to do the conversion with bisulfite in the conventional way, you would lose 70% of your material, your starting material. So as you can see, then if you're losing 70% out of your 3 copies, you're left with nothing in here. There are other problems in terms of cancer and its heterogeneity itself, because especially in the early stages, the cancer patient and cancer patients are presenting really different profiles. So it's not enough to detect one marker, to have a very accurate and analytically specific test only for one marker. So multiplexing really comes into play in here. So more the better, because the disease itself is very complex. So it poses again certain limitations for qPCR, for ddPCR usages. Methodologies that don't use conversion, of course, are frequently used as well, like, for example, methylation sensitive restriction enzymes. But in there, you are limited in having the presence of the restriction enzyme cut sites. So most of the targets, or 40 to 50% of the targets that you're interested in, you just wouldn't manage to target, and you just don't manage to cut with the restriction enzymes. So there are a lot of challenges in designing an assay and designing a methodology for methylation detection, for fragmentation detection, combining this into one and multiplexing into a high level. And this is where the Twist Biosciences solution comes really handy. And we have been working with Twist for 2 years, more than 2 years now on this solution. And so their probes and the option offered is really allowing for high multiplexing. So it allows for capturing both the methylated and unmethylated regions, which is really relevant in early detection of cancer. Due to the mismatch tolerance in the probes, you can capture not only the fully methylated, unmethylated probes, but also the patterns of different methylation stages nicely enriched for. The design itself is really maximizing for the on-bait enrichment, so eliminating any unspecific binding. The chemistry is allowing for control over this. And so in the end, it's really, at least in our experience, very appropriate for low input cell-free DNA analysis, or in this case, an even lower input tumor DNA analysis. In our current panel, we are covering more than 800,000 base pairs with these probes, and which makes it into more than 4,000 target regions. And in our analytical evaluation, we have more than 85%, normally more than 90% of the bases that are on bait. The full data penalty, so that's the uniformity of capture of each of these probes, is really high on less than 1.5, which means that regardless of the region, regardless of the CpG content, regardless of the location of the region, the capture is really uniform over the different regions that we have. So you can see in here on the slide as well that there are several steps for controlling for the on-bait and off-bait capture. You can play around with different hybridization times and temperatures to allow for the very specific and very accurate capture and enrichment in here. So 10 nanograms of cell-free DNA is not a problem to use in this context. Even less than 10 nanograms would work. So it's really appropriate for enriching many targets and in a very accurate and uniform format in here. So from our analytical analysis, one of the good and I would say even more important parts is that what we can see compared to the conventional analysis is that the profiles after the whole procedure of conversion and the capture maintain the original fragment size and concentration profiles, as you would see from just cell-free DNA without going into methylation analysis. What I mean by that is that in a normal cell-free DNA, you would normally see fragments that are wrapping around one nucleosome, but also to 2, 3, or up to 4 nucleosomes. And this is something that with the conventional method you wouldn't see. You would see wrapping around up to 2 nucleosomes, but as the methodology is actually degrading the sample itself to shorter fragments, you don't get the real biological size anymore. Whereas in the approach that we are using, you actually see the original wrapping and the fragment sizes are more accurate to the biological size itself. And on the right side in here, you would also see that the red in here is the conventional method, and the blue is the UDX twist method, where the exact same samples were prepared with the two methodologies and then sequenced, and you can appreciate that in the beginning, the raw data was on the exact same, similar level. So coming out of the sequencer, having the optical duplicates removed, and the values are still quite comparable. But then when we go into identifying the actual PCR duplicates, the fragments, and filtering by quality, you can see that the UDX twist method ends up having significantly more unique reads and more reads that are on bait and with higher depth and per sample per region. So it really gives you more signal, which is just so relevant in the early detection of cancer. Further on in this particular slide, you can see we were comparing duplicate analysis of from cancer patients and from control patients. So the same regions that I already mentioned, both hyper and hypomethylated regions were evaluated, and the correlation between the replicates. So in all total of 49 cancer patients and 45 controls, the correlation was on almost 1. So as perfect as you could get in here. With a minimum correlation of 98.9, basically. Meaning that the methodology is robust, the methylation signal itself is very robust as well, even if you measure the same patient twice in 2 different analyses. So going into case study from here that we have done and using this methodology, as mentioned in the beginning by John, then we are working more on GI cancers, so mostly in colorectal cancer. So this is a study that was done on a case control cohort of colorectal cancers and controls, using 4 milliliters of plasma as a starting material, separating the sample set into training and validation set, where in the training set we had 34 cancer cases and 34 controls. Controls mean that they were colonoscopy checked controls. And a validation set then was extended by including also benign diseases like gastrointestinal diseases, so GID, non-advanced adenomas, the NAA and HB being hyperplastic polyp patients in here. The training set was cohorts from Spain and Ukraine, whereas the validation set also included the German population. So we performed the twist capture methodology, so on our targets, did the sequencing, but of course it's not enough just to sequence and just to capture. It is very relevant because it helps us to go further and to identify the fragments that we are interested in looking at in cancer context. Let's say post-sequencing analysis for us means that we have identified what are the cancer-related methylation patterns, fragmentation patterns. So we have developed our own proprietary algorithm to score this, to score each region and in each sample, identify how many of the fragments from the total are cancer-related. And then combining this into a model which was trained on a training set and then applied on the validation set. And by using the methodology, by using our post-sequencing analysis and scoring and machine learning, we managed to get on a validation set a very nice accuracy values for colorectal cancer detection, especially for the early stage. So the stage 1, I have to apologize that the stages have been a bit smudged together in here, but the leftmost pillar on the sensitivity side, the red side, is the stage 1 sensitivity, which was 83%, and going further on stage 2 at 92%, stage 3 at 92%, and of course stage 4, which is the easiest to detect at 100%. Important part in here is from the specificity side as well, because high specificity in the end of the day means that you are leading less less healthy people to the colonoscopy and follow-up, which is really important for the patient itself as well as for the health system. So the 97% specificity, combining the colonoscopy negative patients with hypoplastic polyp who are considered as false positive if detected, the gastrointestinal disease patients and non-advanced adenoma patients all show the very high specificity here, meaning that the methodology really allows for cancer-specific signal detection, which is not influenced by inflammation or any other confounders in here. The cancer sensitivity on both the proximal and distal cancer was significantly high, both for proximal and distal. So proximal cancers are always a problematic ones to be detected, but in our case, 91% sensitivity could be seen. And from the specificity side, a high specificity could be observed even in the oldest age group. As you know, then methylation signals actually do change with age. So this is a really important distinction that our methods and our methodology and the markers are not detecting age, but they actually do detect the disease again. And as we did this analysis on 3 different populations, then of course we also compared the sensitivity per the collection site, as the specificity remained really high. There was a bit of a drop on German population, but it was also a very small population of 5 patients only of cancer patients. This statistically, as you can appreciate, could be on either way, on 80% or could be also high, 100%. The individual markers that we are measuring are highly correlated to pathways connected to cancer, so signaling pathways, the pathways in cancer, RAP1 signaling pathways. And I'm bringing out here one of the markers that has been shown also previously to be tumor suppressor in colorectal cancer. And the box plots in here are actually showing the difference in methylation signals for 2 separate markers. So the red side is control patients, and the blue side are colorectal cancer patients in here. So you can see that the methylation signals are after the scoring and fragment determination are really distinctly different in here. But going into further to earlier than just cancer detection, then we have made our study also on advanced adenomas. So that's where we are talking about cancer prevention rather than early detection, because detecting adenoma means that you can remove it and you can prevent cancer even manifesting. So we are talking about even smaller percentages of tumor-derived DNA in cancer, so even more need for accurate and analytically sensitive methodology. We performed a similar analysis, so using the hybrid capture methodology, using our algorithms for fragment detection and scoring, and the sensitivity and specificity values you can see in here. So sensitivity of 55%, the specificity on 90% is really something we are very proud about, because as said, this is a pre-cancer, this is a really low shedder, this is a very hard to detect condition. So having a 55% in plot-based test is already meaning that you're making a huge contribution into sending the right people to the colonoscopy here. And the sensitivity is remaining high for all over the different histological subtypes and different grades and combination of advanced adenomas. And when we did the pathway analysis, then once again, the main markers in here are connected to the cancer, the signaling pathways, so highly linked to the cancer development and processes itself as well. So key takeaways from here is that, well, of course, liquid biopsies is very crucial for the cancer detection, but high sensitivity is needed. Multiplexing capability is needed because of the heterogeneous condition that cancer is, especially in the early stages. The hybrid capture-based methodology really allows for the high multiplexing that is needed, for the target enrichment that is needed for both methylated and unmethylated regions. And it also allows for this accurate fragment size estimation. And in our hands with the methodology, we were able to detect early cancers, so early colorectal cancers with really high sensitivity of 83% and for stage 1 and 92% for stage 2, and even more the pre-cancer advanced adenoma detection was also possible with 55% sensitivity, giving this methodology really the hope and boost for helping towards making cancer a curable disease in here. So I would like to also thank all of our contributors, so Twist Biosciences, of course, Diagonal for helping us on the lab side, Gene Planet on the data side, University Polytechnic Catalunya in Barcelona for the data science part, Imperial College on their advice, all of our clinics and hospitals that contributed with the patients. And of course, special thanks goes to our advisors, James Kinross and Michael Roll, who have given their good thoughts on how to set this up and how to work this towards clinical use. So thank you, and I'm open for any questions.
And once again, a big thank you to Christy for presenting during this webinar today. And I want to thank everyone for joining us today. Have a great day. Until next time.
Thank you.